Proteins, Part 4. Biosynthesis of amino acid. In this e-lecture, we will study the biosynthesis of amino acid of oxaloacetate precursor family. Let us start. All amino acids are synthesized by transamination of keto acid, and these keto acids are derived from the intermediates of glycolytic pathway, pentose phosphate pathway, and tricarboxylic acid cycle. In this picture, which shows the precursors of amino acid synthesis, we see that the ribose 5 phosphate, which is an intermediate of pentose phosphate pathway, give rise to histidine. Next, 3-phosphoglycerate, which is an intermediate of glycolytic pathway, gave rise to serine, and then serine gave rise to glycine and cysteine. Phosphoenol pyruvate along with the erythrose 4-phosphate give rise to tryptophan, phenylalanine and tyrosine. Pyruvate, another important metabolic intermediate of glycolytic pathway, give rise to alanine, valine and leucine. Alpha ketoglutarate, which is a metabolic intermediate of Krebs cycle or tricarboxylic acid cycle, give rise to glutamate, and then glutamate give rise to glutamine, proline and arginine. The last one is the oxaloacetate. Oxaloacetate give rise to aspartate, and then aspartate give rise to asparagine, methionine, threonine, lysine and isoleucine. Thus we see that all the amino acids are derived from the metabolic intermediates of three important pathway, and these three important pathway are glycolytic pathway, pentose phosphate pathway and tricarboxylic acid cycle. The precursors of amino acids. On the basis of the precursors of amino acid, 6 families are recognized, and these families are oxaloacetate precursor family, pyruvate family, alpha ketoglutarate precursor family, 3-phosphoglycerate precursor family, phosphoenol pyruvate and erythrose 4-phosphate precursor family, and the last one is the ribose 5-phosphate precursor family. In this e-lecture, we will study about the synthesis of oxaloacetate precursor family. As we see that the oxaloacetate first give rise to aspartate, and then this aspartate give rise to other 5 amino acids, and these amino acids are asparagine, methionine, threonine, lysine and isoleucine. Thus, the conversion of oxaloacetate to aspartate is the most important step, which give rise to other 5 amino acids. Let us see this reaction. Now, the synthesis of aspartate from oxaloacetate. The aspartate is derived from oxaloacetate by a simple transamination reaction, and in this transamination reaction, the glutamate donates its amino group to oxaloacetate and aspartate is formed. This reaction is catalyzed by enzyme aminotransferase. The aminotransferase has a prosthetic group pyridoxal phosphate, and this pyridoxal phosphate is abbreviated as a PLP. This pyridoxal phosphate is a derivative of pyridoxine, which is vitamin B6. Aspartate is utilized in the formation of 5 other amino acids, and these are asparagine, methionine, threonine, lysine and isoleucine. Now let us see the synthesis of asparagine from aspartate. The aspartate is first activated by ATP. This process is known as adenylation. One pyrophosphate is released in this reaction and an intermediate is formed. This intermediate acyl adenylate is then converted to asparagine by accepting an amino group from glutamine, and thus asparagine is synthesized. The donor of amino group is glutamine and one AMP is released in this reaction. Now, aspartate also gives rise to methionine. In this reaction, this synthesis of methionine is completed in 7 steps. 1, 2, 3, 4, 5, 6, 7. 7-step reaction. First step is aspartate give rise to aspartyl beta phosphate. The biosynthesis of methionine starts with the activation of aspartate by ATP. And this activation of aspartate occurs in the presence of enzyme aspartokinase. And the product form is aspartyl beta phosphate. This activated aspartyl beta phosphate is reduced by the enzyme aspartyl beta semi-aldehyde dehydrogenase at the expense of NADPH plus H plus, and this results in the formation of aspartyl beta semi-aldehyde. The aspartyl beta semi-aldehyde is an important intermediate as it gives rise to threonine on one side, lysine on another side, and on third side it gives rise to methionine. The aspartyl beta semi-aldehyde is reduced by homoserine dehydrogenase in this third step and NADPH plus H plus. As a result, the homoserine is formed. This homoserine is converted to succinyl homoserine in the step 4th, and this reaction is catalyzed by the enzyme homoserine acyl transferase. The succinyl homoserine in next step 5th, this succinyl group of succinyl homoserine is replaced by cysteine, and this reaction 5th is catalyzed by the enzyme cystathionine gamma synthase. As a result, cystathionine is formed. This reaction is also a PLP dependent. In the next step 6th, this cystathionine beta lyase enzyme liberates pyruvate and ammonia from cystathionine to produce homocysteine. And in the last step 7th, the homocysteine is methylated by the enzyme methionine synthase, and the donor of methyl group is methyl tetrahydrofolate, and this produces methionine and tetrahydrofolate is released. Thus, in 7th step, methionine is formed from aspartate. Now, the biosynthesis of threonine from homoserine. As we see in this reaction, the threonine is formed from homoserine and this homoserine is formed from the aspartate. Now, we see the synthesis of threonine from homoserine by two-step reaction. The homoserine is first phosphorylated by the enzyme homoserine kinase, and 1 ATP is also utilized as a phosphate donor. The intermediate product is phosphohomoserine. In the second step, this phosphohomoserine is converted to threonine, and this reaction is catalyzed by threonine synthase. This enzyme is also a PLP dependent enzyme, and 1-phosphate is released in this reaction. As a result, threonine is formed from homoserine. Now the biosynthesis of lysine. The biosynthesis of lysine also occur from the aspartate, and this is a complicated reaction. It occurs in 3 steps. The initial steps up to the formation of aspartyl beta semialdehyde is the same as we discussed in the methionine synthesis. And starting from the aspartyl beta semialdehyde, the reaction is completed in 3 stages. Stage 1st, stage 2nd, and stage 3rd. In stage 1st, this aspartyl beta semialdehyde is converted to N-succinyl 2-amino 6-ketopamylate. In stage 2nd, N-succinyl 2-amino 6-ketopamylate is converted to 2,6-diaminopamylate and succinate. And in stage 3rd, this 2,6-diaminopamylate is converted to lysine. Now let us see a biosynthesis of lysine. In the stage 1st, this aspartyl beta semialdehyde combines with a molecule of pyruvate and to form a dihydropicolinate. And this reaction is catalyzed by dihydropicolinate synthase enzyme. In the next step, second step, this dihydropicolinate is reduced to piperidine 2,6-dicarboxylate, and the reaction is catalyzed by the enzyme piperidine 2,6-dicarboxylate dehydrogenase enzyme. In the third step, this N6 link, N nitrogen and carbon 6 carbon link in the ring of piperidine is hydrolyzed, and succinyl group is transferred to the nitrogen by succinyl CoA. This results in the formation of N-succinyl 2-amino 6-ketopamylate, and the reaction is catalyzed by the N-succinyl 2-amino 6-ketopamylate synthase enzyme. Thus, in stage 1st, we see that aspartyl beta semialdehyde is converted to N-succinyl 2-amino 6-ketopamylate. Stage 2nd, this N-succinyl 2-amino 6-ketopamylate is converted to 2,6-diaminopamylate by 2-step reaction. In step 1st, an amino group is donated by the glutamate to the 6th carbon of N-succinyl 2-amino 6-ketopamylate. This is a simple transamination reaction, and this reaction is catalyzed by the enzyme succinyl diaminopamylate aminotransferase. It is a PLP dependent enzyme, and as a result N-succinyl 2,6-diaminopamylate is formed. The N-succinyl 2,6-diaminopamylate is converted to 2,6-diaminopamylate as succinate group is hydrolyzed by the enzyme succinyl diaminopamylate desuccinylase, As a result, 2,6-diaminopalmilate is formed and succinate group is released. In stage third, this 2,6-diaminopalmilate is converted to lysine by two-step reaction. In step first, diaminopalmilate epimerase enzyme introduces an isomeric change and it produces the mesoform of mesodiaminopalmilate. This mesoform is then decarboxylated by enzyme diaminopalmilate decarboxylate, converting this mesoform into lysine. This enzyme is also a PLP dependent enzyme. And in this way, lysine is formed from aspartyl beta semialdehyde by in 3 stages. Now the biosynthesis of isoleucine. The biosynthesis of isoleucine starts with the threonine. In step first, this threonine is converted to alpha ketobutyrate by a PLP dependent enzyme threonine hydratase, and one ammonia group is released in this reaction. The alpha ketobutyrate then combines with the pyruvate TPP complex to form alpha hydroxybutyrate. This reaction second is catalyzed by the enzyme acetolactate synthase. In the third step, the ethyl group is shifted to the carbonyl carbon of the acetyl group, and an alpha and beta carbons are reduced by the formation of an attached hydroxyl group. And the enzyme involved is acetohydroxy acid isomer reductase. As a result, alpha beta dihydroxy beta methyl valerate is formed. NADPH plus H plus is also used as a reductant in this reaction third. In the next step fourth, this alpha beta dihydroxy beta methyl valerate removes a molecule of water to produce alpha keto beta methyl valerate. The alpha keto group of this alpha keto beta methyl valerate is now transaminated by glutamate in step 5 to produce the isoleucine. The donor of the amino group is glutamate and an alpha ketoglutarate is released in this reaction. This is a simple transamination reaction and PLP dependent. The reaction is catalyzed by the enzyme valine aminotransferase. As a result, isoleucine is synthesized. Thus, we see that 6 amino acids are synthesized from oxaloacetate, and oxaloacetate precursor family is very important family. Now, let us summarize all these reactions and biosynthesis of amino acids. We have seen this family, oxaloacetate precursor family, and we have seen that the oxaloacetate first of all give rise to aspartate by simple transamination reaction, and then aspartate give rise to 5 other amino acids, and these are asparagine, methionine, threonine, lysine and isoleucine. The isoleucine is formed from threonine. The aspartate is formed from oxaloacetate by a simple transamination reaction. This is very important reaction as the aspartate formed give rise to 5 other amino acids. First, the formation of asparagine from aspartate. and the aspartate give rise to methionine by 7-step reaction. And in this 7th step, we have also seen that this aspartyl beta semialdehyde is an important intermediate. And this aspartyl beta semialdehyde give rise to threonine and lysine and methionine. Now in this 7-step reaction, the aspartate give rise to methionine. Homoserine, which is an intermediate, give rise to threonine. Biosynthesis of lysine completed in 3 stages. And the biosynthesis of lysine also occur from aspartyl beta semialdehyde, which is an intermediate in the biosynthesis of methionine. In the stage first, aspartyl beta semialdehyde give rise to N-succinyl 2-amino 6-ketopalmilate. In stage second, N-succinyl 2-amino 6-ketopalmilate give rise to 2,6-diaminopalmilate. And in stage third, 2,6-diaminopalmilate give rise to lysine. Now the biosynthesis of lysine occur from threonine. In this way, we see that the oxaloacetate precursor family, and in this family 6 amino acids are synthesized. Now this image shows the biosynthetic pathway for the synthesis of oxaloacetate derived family, and in this picture we clearly shows that the oxaloacetate give rise to aspartate, aspartate give rise to asparagine. Aspartate also give rise to aspartyl beta semialdehyde, which is an important intermediate, as it give rise to homoserine on one side, and homoserine give rise to threonine and methionine on another side. This aspartyl beta semialdehyde, when combines with pyruvate, give rise to lysine in 3 stages reaction. Threonine then give rise to isoleucine. Thus, we see that oxaloacetate is a precursor family, is very important precursor family, and 6 amino acids are synthesized in this synthesized from oxaloacetate. For further readings, I would like to suggest a book of biochemistry by S.N. Gupta. I hope you like this video and get benefit of it. Thank you for watching.